Our post-coffee break keynote speaker is Dr. Pete Lollar, who is a Hemophilia of Georgia professor of pediatrics and director of hemostasis research in the Aflac Cancer and Blood Disorders Center in the Department of Pediatrics at Emory University in Atlanta. He has had a long and outstanding career in hemostasis research. His interest is in the structure and function of factor VIII von Willebrand factor complex, the immune response to factor VIII, and the biosynthesis of factor VIII. This expertise has led to his invention of Obejur, which is a recombinant therapeutic used to treat hemophilia A patients with acquired factor VIII inhibitors. So, like many of us here who are trying to save lives, he has accomplished this in the development of therapeutic. More recently, he is also an inventor of ET3, a factor VIII transgene used in lentiviral-mediated hematopoietic stem cell gene therapy to cure hemophilia A. Having known Pete since 1987, I am well aware he's very creative with eclectic hobbies such as math, fine arts, and he's an accomplished fly fisher, fly tyer, and Delta blues guitarist. I'm sure that if Earl Davie was here right with us, he'd be pleased that Pete was selected to deliver this keynote address. Dr. Lollar.
Wow, that was quite the introduction. So it's a real honor to be invited to give this presentation at the Earl Davie Symposium. I met Earl on several occasions, first when I was a hematology fellow, and then when I was a young assistant professor. And I can just say that he was a perfect gentleman and a pleasure to be around. He published a lot of papers in the field of hemostasis, and some of them have been directly relevant to the research that I've done over my career. His laboratory was the first laboratory to purify factor VIII to homogeneity, and also he did foundational work in the structure and function of von Willebrand factor, both biochemistry and also in the cloning of the human factor VIII cDNA. It's also notable that two of the people listed on these publications, Gordon Vihar and Evan Sadler, trainees in his laboratory, went on to become giants in the field of hemostasis research and biotechnology. So what I'm going to describe today is our studies on the conformation of von Willebrand factor, which represents a digression, a serendipitous digression from what we were initially studying, which is the immune response to factor VIII, which is a major problem in the management of patients with hemophilia A. And our hypothesis was that immune complexes play a role in the immunogenicity of factor VIII. And because factor VIII is bound to von Willebrand factor, which has a lot of binding sites for factor VIII, and because IgG, the primary antibody in the immune response to factor VIII, is bivalent for factor VIII, you potentially can form very large complexes. So our idea was to do physical characterization of these complexes as initial work in studying the potential immune response of factor VIII to immune complexes. Some of the methods that are used to characterize really big complexes are shown here. The ones in red, analytical ultracentrifugation, multi-angle light scattering, and dynamic light scattering are ones that I'll be discussing today. The von Willebrand factor, I think many of you here are familiar with this structure of von Willebrand factor. It has a subunit that has a molecular weight of about 280 kilodaltons, and the subunit is made up of small domains of 20 to 40 kilodaltons. There's X-ray structures of many of these domains. They're typical globulars, quasi-spherical particles that form the VWF subunit. And then VWF subunits self-associate at the C-terminal ends by disulfide bonding to form the dimer, and then VWF dimers then associate through N-terminal disulfide linking to form multimers. And interestingly, the cells, when they're making von Willebrand factor, they release them after various links have been synthesized. So von Willebrand factor is intrinsically heterogeneous. The molecular weights in plasma range from about 500,000 all the way up to molecular weights that haven't been measured, but probably over 10 megadaltons, 10 million daltons. So the conformation of von Willebrand factor has been studied primarily by electron microscopy, but also internal reflection fluorescence microscopy and atomic force microscopy. And these images, especially the EM images, have led to this descriptive characterization of VWF as having a ball of yarn conformation or a tangled coil, or my favorite is a compact fuzzball, which implies that there are interactions within the subunits that keep it bound together, and it circulates in this globular, almost spherical form. So this has led to what is considered the established mechanism for the initial stages of activation of von Willebrand factor, which floats in an inactive form in plasma in this compact fuzzball form under laminar flow, and then during disruption of the blood vessel, this coil unfolds, and then there are subsequent activation events, and VWF participates in its function in platelet adhesion, platelet aggregation. So the first thing that we wanted to do to characterize immune complexes was to leave out the antibody and start and do some characterization of the factor VIII VWF complex. And we isolated VWF, human VWF, from alpha-NATE, which is a therapeutic product that's used in the management of patients with hemophilia and von Willebrand disease. And what this does is remove the human serum albumin, which has been added back to alpha-NATE as a stabilizer. And when you do this, you get a very clean preparation of the factor VIII von Willebrand factor complex. And importantly, the ratio of factor VIII to VWF in native plasma is really low, only about 1 to 5% of the mass of a VWF factor VIII complex is VWF. So when you're looking at the physical properties of a factor VIII VWF complex, you're just basically looking at VWF. So the initial thing we did to characterize this complex was to perform analytical ultracentrifugation. And in this method, what you do is you put a sample purified protein into a cell that has transparent windows, and then you put the cell on a rotor and you subject it to centrifugation. And there is an optical system in the ultracentrifuge. There's actually two of them, but the one I'll discuss today is an absorbance optical system. And you measure the A280 of the sample as a function of the radial position in the cell. So as the protein starts to sediment towards the bottom, you can follow how the cell it clears the protein as it becomes pelleted. So this is what AUC of these unfractionated VWF complexes looks like. On this graph here is the raw data. What you see on the x-axis is the radial position, and then on the y-axis is the A280. And then these curves are a series of scans which were taken at 4-minute intervals. And you see that there are these curves form what's called a boundary. And this boundary is sedimenting towards the bottom of the centrifuge cell with time. And this is how sedimentation coefficients are determined. Like if you've ever heard of 16S-RNA or an AES ribosome, that was done in an analytical ultracentrifuge. So with VWF, what you would expect is not just a single sedimentation coefficient because it's heterogeneous. And that's what you see here. In addition to the data in this graph, there's also a fit of the data to a model which provides the sedimentation coefficient distribution in the preparation. And that's shown here on the x-axis is a sedimentation coefficient. And you can see there's this large peak around 20S, but there are also species in there ranging from down from 5S to 60S. And if you integrate this peak, you get a hard physical number called the weight average sedimentation coefficient. And for this sample, it's 21S. So this is what we expected and was not surprising. However, when we repeated this study by loading increasing concentrations of VWF factor VIII complexes into the centrifuge, what we found is the sedimentation coefficient decreased with increasing concentration. And that's not supposed to happen with compact globular proteins. And as a control, you can see that bovine serum albumin up to 1 mg per mil does not show a change in its sedimentation coefficient. And this kind of suggests that maybe the VWF multimers are getting tangled up, loosely speaking, with one another and having a hard time sedimenting because of the fact that they're getting tangled up. So we decided to pursue the physical characterization of VWF multimers in more detail. And to do this, we fractionated VWF by size exclusion chromatography. And we collected, in this case, 13 fractions, which are numbered on this graph. Shown down on the bottom is SDS agarose gel electrophoresis, showing that we fractionated the multimers from higher molecular weight to lower molecular weight. None of these samples is homogeneous in the sense that there are several multimers in each sample. That turns out really not to be a big problem when you're doing characterization, physical characterization of polymers, as I'll discuss in a few minutes. So the first thing we wanted to do is get better shape information from these samples. And the way we did that initially was to measure the frictional coefficient of von Willebrand factor. Molecules in solution, as they're moving through solution, they feel a resistive force, which is a frictional force. And that force is described by the frictional coefficient. The frictional coefficient of a sphere is related to its radius by Stokes' law. If you actually experimentally measure the frictional coefficient, which I'll show you how we do that in a minute or two, what one gets is a radius which is called the hydrodynamic radius, which is this analog to Stokes' law. So you measure the frictional coefficient experimentally, and then you ask the question, well, what would the frictional coefficient be if it were a sphere? If I know the mass of a particle and I know its density and it's a sphere, I can calculate its radius. In fact, if there's any high school students doing work in your laboratory, give them that test when you get back to the lab, because it's an easy test. So for a protein, we know the mass if we know the molecular weight, and we know the density from the amino acid and carbohydrate composition. So for von Willebrand factor, we can calculate this hypothetical radius, and then we can compare it to the true radius. If it's a sphere, they should be the same, and this ratio should be 1.0. So the frictional ratios of hundreds of proteins have been measured. Here's some examples of globular proteins, and the frictional ratios tend to be close to 1.0. Bovine serum albumin, these are X-ray structures, is highly non-spherical. Its frictional ratio starts to increase a little bit. The frictional ratios of some coagulation proteins have been measured by Dr. Prysdale for factor V-A and factor Xa. This is a well-established way to characterize, get some shape information about proteins. Fibrinogen is highly asymmetric. It has a frictional ratio of 2.3. So the way the frictional coefficient is measured in most laboratories is to measure the diffusion coefficient, which is related to the frictional coefficient by the Einstein diffusion equation. It's simply inversely proportional to that. And what Einstein also proposed, if I can get this to work, is that diffusion itself is the same phenomenon that had been previously observed in the microscope called Brownian motion. Okay, so these are particles undergoing Brownian motion, and if you were to trace the path of any one of these particles, they undergo this wild, random walk. As Einstein said, they alter their positions in the most irregular manner possible. And in the middle are some traces of a particle undergoing Brownian motion, and they look strangely similar to the electron micrograph of von Willebrand factor, which I'll return to, and is one of the main take-home points of this presentation. So the diffusion coefficient is commonly measured by dynamic light scattering. And the way this works is you shine light on a mixture of molecules, and the light interacts with the molecules, and they actually produce light waves themselves, which go out in all directions. And that's kind of the 10-word definition of light scattering. So early on, the molecules are still in their same position, and well, let me back up a second. When they are producing these light waves, the light waves can interfere with each other, so the intensity that's actually observed is fluctuating a little bit. Early on, the molecules are still relatively in the same positions, so this intensity has not changed much. But as they undergo Brownian motion, they become completely unrelated to where they were before with respect to the other molecules. So this correlation of intensity starts to decrease. So what is measured in a dynamic light scattering instrument is this decay in what's called the autocorrelation with time. So for these fractionated VWF multimers, we did that. We got really nice light scattering data. What I'm showing is a series of curves for the 13 different samples that we had. You can see this decay in autocorrelation with time. There is a theory which then allows you to get the diffusion coefficient from this decay, and technically what it produces is what's called the Z-average diffusion coefficient. So we got the diffusion coefficient, and now we can calculate the frictional coefficient, but we still need to get this frictional coefficient of the hypothetical sphere, and we need the molecular weight for that. So to do that, we measured the molecular weight of these samples by multi-angle light scattering, another light scattering phenomenon. And in this case, you shine light on the sample and you measure the time average light scattering as a function of the scattering angle. And the theory predicts that it produces a hard physical constant, the weight average molecular weight, and it also produces for larger particles the radius of gyration, which will become important in a couple of minutes. So we used a Wyatt size exclusion chromatography MAL system to do this. Von Willebrand factor was pumped through an SEC column. It goes through a multi-angle light scattering detector, and then also a differential refractometer, which allows one to estimate its concentration, which is required for the molecular weight calculation. So with all that being said, we get a frictional ratio, and for von Willebrand factor, for these fractions, they ranged in molecular weight from about 1.7 megadaltons to 5 megadaltons, and the frictional ratios were 2.7 to 3.2, very high. So it's really clear now that von Willebrand factor, the dogma that von Willebrand factor is a condensed particle, and it's part of the activation process of VWF involves unraveling this coil. I think it's clear that this is incorrect. So VWF is not a ball of yarn. It's not a fuzz ball, but what is it? So if you take a step back and just talk about conformations of polymers, if you pick up a polymer chemistry book, which I wouldn't necessarily advise, they describe a spectrum of conformations, ranging from a globule where a linear polymer is collapsed in among itself because the components of the polymer really don't like the solvent as much as they like interacting with themselves. And at the other end is a rod conformation in which the polymer has negative or positive repulsive forces which extend it into a chain. And in between that in this spectrum is a random coil. And for VWF, we can envision that the globular components which make up a subunit are strung along like beads on a chain, and each of these monomers is undergoing a random walk, but it's restricted. It's a restricted random walk because it's connected in the chain. This is a picture from a polymer chemistry book, which just shows a single confirmation of a computer simulation of what a random coil would look like, but you have to imagine that this thing is moving around in solution in all manners of conformation possible. Then there is what's called the worm-like chain, which is intermediate between a random coil and a rod. This indicates that the polymer has some degree of stiffness, and the polymer chemists like to use a garden hose as an example, or as an analogy, in that if you have a garden hose and you just throw it on the ground, the two ends are really kind of uncorrelated with respect to one another. But over a short segment of the chain, 6 inches or so, there is a stiffness because of local conformation. So the theory of polymer conformations goes back a long time. Paul Flory got the Nobel Prize in 1974 for his contributions in this area. And what he developed was a theory of the molecular weight dependence of the sedimentation coefficient, the diffusion coefficient, and the radius of gyration and the intrinsic viscosity for this spectrum of polymers: spheres, random coils, and rods. And so we can measure, we did measure the sedimentation coefficient, diffusion coefficient, and the radius of gyration, so we could actually look at his theory for VWF multimers. Now, the radius of gyration, which comes out of a multi-angle light scattering measurement, is a weighted average of the distribution of mass elements of the particle around the center of mass. And it's another hard physical parameter which can be determined by both multi-angle light scattering and, for smaller particles, SACs and small angle neutron scattering. So these are multi-angle light scattering plots, Berry plots of VWF. On the left graph is the scattering angle, or a function of the scattering angle, and on the y-axis is this function of the scattering intensity. And what you see for these 13 fractions is a very nice linear Berry plot. As you see, the angle increases, this light scattering increases, and the slope of the line gives you the radius of gyration, and the y-intercept is a function of the molecular weight. So that's how that SEC-MOS is used to determine molecular weight and radius of gyration. On the right graph, I'm showing this plot of radius of gyration versus molecular weight. So this kind of information, light scattering, AUC for sedimentation coefficient, and DLS for the diffusion coefficient, allows one to construct what are called conformation plots in polymer chemistry. And Flory theory predicts that there's a linear relationship between the molecular weight and these physical parameters. And what we found with these 13 fractions is that there is indeed a linear relationship. This relationship is on a double logarithmic scale. And then Flory theory predicts what the slope should be, whether it's a sphere, a random coil, or a rod. And what we found is that the slopes of these plots are quite consistent with random coil behavior. And trying to get this, there we go. This is just a computer simulation of a random coil, identical beads on a string. This is what it would look like in solution. And again, it looks a lot like what von Willebrand factor looks like if you take this random coil and just drop it onto an EM grid. Finally, we looked at whether von Willebrand factor multimers have any properties of a worm-like chain. And there's a model for this in which the parameters of the chain are the contour length, which is just the length of the chain completely stretched out, the diameter, which would be the diameter of the garden hose here, and then the persistence length, which is a measure of the stiffness of the chain. The more stiff the chain is, the longer the persistence length. So that these LP ratios predict are either small or large, whether the chain is stiff or flexible. And these can be modeled experimentally into experimental parameters, the radius of gyration and the hydrodynamic radius, which we can measure. And then the ratio of the radius of gyration and the hydrodynamic radius can be used to examine the stiffness of the chain. So what we found, surprisingly, is that this RG-RH ratio for these samples ranged from about 1.3 to 1.5, and that is in the range for an LP ratio for a very flexible worm as opposed to a worm that's stiff, which to me was surprising because it's hard for me to kind of imagine how even these globular domains don't have some degree of stiffness as they're connected to one another. But that's what we found, and we're interested in exploring that further. So in summary, we studied the conformation of VWF using standard methods of polymer chemistry. This has been done under static non-flowing conditions, and it displays random coil behavior. We think that VWF is perhaps the most well understood, documented example of a protein existing as a random coil under native conditions, and that VWF has surprising flexibility, and that models of the activation and biosynthesis of VWF should start with consideration that it's a random coil when it's in its native conformation. Thank you.
Thank you very much. Other questions?
Great talk. Question here. May I know, would the structure of VWF affect platelet binding?
Can you repeat that question for me, Eddie?
I didn't get that. Could you say it again?
Would the structure of the VWF polymer, either it's coil or rod, would that affect platelet binding?
How does the random coil affect platelet binding?
Don't know. Have not tried to correlate the conformation of VWF to bioassays.
Yeah, so we also study FVIII VWF. So in our experiment, we add in, for example, we bind FVIII on VWF multimer, and we found the final binding ratio under static no flow condition is about 0.8 to 0.9 with FVIII per VWF monomer. So my question is, based on your random coil structure, I just want to know whether you can explain how can we get such a high binding ratio between FVIII to VWF multimer? Because FVIII is such a big molecule. The second question actually is still follow this trend. We never see the 1-to-1 ratio, then FVIII to one FVIII to one VWF monomer. So I just want to know, do you have some idea about why we cannot reach 1-to-1 ratio?
Okay, so
thank you.
If I understand your question correctly, if you look at do an assay of VWF in plasma and you say, well, there's 1 unit per mil of factor VIII and 1 unit per mil of von Willebrand factor, that doesn't mean that the ratio on a molecular basis is 1-to-1. If you take von Willebrand factor and take away the factor VIII, you can add factor VIII back and completely saturate all the subunits. But in vivo, I think it's well known that the average VWF molecule floating around does not have a single factor VIII bound to it. And the ratio of factor VIII to VWF on a molar basis is per subunit of VWF in plasma is about 1 to 50. And the molecular weight of factor VIII is very close to the molecular weight of the VWF subunit. So when you're looking at, in a physical study, you're looking at the mass, some measurement of the mass of the particle, and you're looking at a factor VIII VWF complexes, which has been purified from a therapeutic concentrate. So it's a physiological complex. You're basically just looking at VWF. Although one of the things that we would still want to do is purify VWF and take away the factor VIII and just study VWF in the absence of factor VIII. We haven't done that yet.
Great talk, thank you. Great talk. Two quick questions. When you did the DLS, how did you select different populations? Did you see different populations or always have one population?
So the curves I showed, those are size fractionated complexes. And when I showed the SDS agarose gel, you could see that each one of the fractions had more than one multimer in it. The curves I showed, each individual curve was from a single SEC fraction. What happens when you have a sample, which is what polymer chemists would call polydisperse, is that you still are able to measure a diffusion coefficient, but it's an average diffusion coefficient of all of the species that are in there. And technically it's called a Z-average. And the Z-average and the weight average sedimentation coefficient and Z-average diffusion coefficient, they actually work out to produce a weight average molecular weight if you want to do that calculation. And it's all part of the algebra in polymer chemistry where you have to say, okay, I've got more than one species in here, I've got to do an average. And the theory underlying DLS produces, it's well established that what you're getting when you fit that plot, I showed you fitted curves, is you get an average diffusion coefficient, which is technically Z-average, which is a molecular entity. You can look up and see how that is defined with respect to the concentrations of the polymers that are in the sample.
Thank you. Can I ask a second question? The samples you use, were they pool samples from one donor, or was multiple donors, or just from one?
They're from multiple donors because alpha-NAT is a plasma-derived, thank you for pointing that out. That's a plasma-derived product which is collected from multiple donors. They pool the plasma, and then it goes through a purification procedure. They actually get highly purified factor VIII VWF complexes, and then they add back human albumin as not an impurity, but as a stabilizing agent. So the material really only has three proteins in it: factor VIII, albumin, and VWF from multiple donors.
Hi, great talk. I was wondering a little bit about what may happen to this random coil structure under shear flow, specifically in regards to cleavage by atom TS13 and also VWF self-association.
I think that's a really great question. And there has been some work done on random coils under shear conditions with DNA. I should also point out that a lot of polymer chemistry is established in the organic chemistry field because these polymers are what makes plastics. I think it would be really interesting to have some sort of experimental approach in which one could follow the unraveling, if you could follow the cleavage of a random coil polymer under conditions of flow. The methods that we're using are really straightforward, and I think to look at the random coils as they align in a flow field is an area that's been actively studied. I'm not a polymer chemist. I think this would be an area of interest of polymer chemists, but the ability of random coils to align in a flow field and how that would affect their susceptibility to proteolysis and access to atom TS13, I think would be an interesting area of investigation.
Thank you.
All right, Jose?
Hi, thanks. That was a great talk. So I'm just curious about the structure of you say you have globules within the random coil. Do you have any idea what those are? Could it be a bunch of internal domains surrounded by flexible domains, or are they individual domains surrounded by flexible domains? Do you have any clue?
Yes, we do. It was a question we were really trying to address, whether we could discern any type of interactions which were potentially collapsing the big long VWF multimer into something where there is an intramolecular interaction. The dogma is that there's a lot, and that the VWF is floating around as this coil or fuzzball. And the last slide I showed, where we were looking at trying to look at the flexibility of VWF, it's consistent with VWF being the analogy that Flory actually used was a string of pearls. And here we have VWF in which the globular domains, they're not identical pearls. You know, we've got A1, A2, A3, but still there are these spherical clusters clustered along the chain. And what we found in that last slide is that VWF appears like there's absolutely no interaction between these domains from one subunit to another, and that there's actually flexibility between, let's say, the A1 and A2 domains, so that the thing is really just a loose coil with no evidence of stiffness that we can find whatsoever.
Thank you. Pete? Clint had a question for you, but in the sake of time, he's deferred to conversation later, and there's about half a dozen questions here online as well, which is pretty impressive. I'm going to ask you one for online because of the sake of time, from Dr. Lillicrap from Queens. He asks, how do you think these structural properties of von Willebrand factor influence the interactions with factor VIII in different parts of the circulation?
I think that VWF being a loose coil, I would predict that factor VIII looks at VWF as a particle that has identical non-interacting sites, and that factor VIII would bind to any component of VWF, any VWF subunit independently and randomly, but that's just a guess.
Thank you very much.